Hello, my name is Oyvind Isaksen. I'm the CEO of poLight. I'm here with Professor Jörg Winter, CEO of PhenoSys. PhenoSys was founded in 2006 and is the leader in innovative technology for animal behavior research. Their Mini-2P turnkey system delivers critical insight into neurological illness such as Alzheimer and dementia. Professor Winter, thanks for joining me today.
Well, thank you, Dr. Isaksen, for inviting me. Now, one reason I'm here is because you decided to adapt consumer market technology, the micro TLens, to the needs of science. And your component, this component, has really become the heart of a high-tech scientific instrument.
Yeah, thanks, Jörg. And to be honest, when we decided to support this initiative, started with Kavli, it was really something which was a very motivating thing for us, that we were able as a company, commercial company, not only chasing the profit and the gross margin and what have you, but also into different industrial and commercial instrumentation and products, but also being able with this technology to provide important contribution to important research. This was really something which was super motivating for us. My mother is actually having dementia, and it's so sad to see that there's so little they can do. So the more we can, in a way, support in that direction, the more I'll get more motivated to develop new technology.
Mm-hmm. Well, it's personal experiences like the ones you are making yourself that is motivating many scientists too, I can tell you.
Yeah. Okay, I'll ask you to spend a little time talking about the importance of the 3D recording of brain tissue in freely moving animals and the value of the Mini 2P solution and how tunable optics and, of course, in specific TLens is used. So your company is recognized as a leader in unique instrumentation in behavior biology, and you're also the chair of cognitive neurobiology at Humboldt University in Berlin. From your perspective, how important is it for researchers to kind of see this brain activity in active mice?
Yes, the key that you are underlining there, activity, true behavior, is really at the heart of what this new technology is making possible. So to go to an analogy, when you have a heart problem, your cardiologist will put you on an exercise bike or provide you with a 24-hour ECG recorder. And this is because the full functionality of any of our organs, the heart, the brain, can only be assessed when the organ is under full operation. So the brain controls the sensing of environment, what we see, we hear, and so forth, and it controls the actions, the behaviors that we perform. And only when the brain is in this operational mode during behavioral activity can we really study its functionality, or what's equally interesting, its malfunction. And we're using mice for this because they're mammals like us, and they have about the smallest brains in which we can study the same processes that also run in our human brains.
So helping scientists to see, with the help of tunable optics inside the brain, clearly seems like a critical function in this research, and definitely sounds like delivering this level of vision in the truly advancing, the research in this area. Okay, let's talk a little bit about the role of the Mini 2P solution playing in this kind of brain research. Your turnkey system is small, deliver very fast, high-resolution images. And as you know, we were actually fortunate enough to work with the Kavli Institute on the Mini 2P innovation. How, in your opinion, how has this unique device impacted research in this area?
Well, the key is really here the miniaturization. What it does enable is that we obtain signals, the miniaturization of microscopy technology. What it enables is that we see the brain in operation using visual technology rather than than electro technology. And the reason for that is that a few decades ago, scientists developed a method that allowed them to turn the electrical activity that kind of silently governs brain and brain activity into what some people might say a flashing Christmas tree. So whenever a neuron sends information, it also emits a pulse of light. And this has led to an explosion in the development of microscopy technology to watch the brain in action, because now you're just looking for light pulses rather as before for changes in electrical activity. And with former technology, these two-photon microscopes are really big, huge machines, and normal behavior of a little mouse is simply not possible when an animal is under them or connected to them. Now, the Nobel laureate team at the Kavli Institute were the first to realize that the key components of microscopes had now been miniaturized to an extent that it should be possible to build a miniature brain explorer weighing only a couple of grams. And with that belief, and with the help of companies, of course, like yours, they succeeded with this development. And the result now really allows us visualizing connection points between neurons in freely moving around animals. And it's in the dynamics of the plasticity of these brain connection points that allow us to learn and behave sensibly or lose that ability.
Interesting. Just a follow-up question, Jörg, on this. This development of Mini2P, very compact, light, which you say that is actually enabling doing important observation in a more natural circumstance, so to speak, that it can freely move to see the response. So that's clearly something I understand very easily, that must be very important to understand what's really happening. But that miniaturization has also any, would you say, implication of making this kind of advanced instrumentation more accessible for more people or for more labs, or would you say that not really that dimension?
Well, fortunately, this is not always the case that newer technology has the potential of reaching wider use. In this case, it is, because due to the fact also to your company incorporating consumer electronic components in there, the price has actually dropped dramatically compared to the very big two-photon machines that scientists are currently using. And we're talking about a halving, or maybe going to 1/3, 1/4 of the price of what people would have paid originally. So it will be a much larger research community that will be able to afford this technology and use it in their research.
So more people being able to do important research, meaning should create more and quicker results, helping my mother and others.
Definitely, yes.
So Mini2P, we're talking about results. I guess it's still too early to say that Mini2P have kind of resulted in extremely important findings, meaning that we now understand so much more, that we have the tablets or the injection we need to kind of delay or to make people feel better. Is that too early to say?
Well, I think we can already say that we can now see details in the freely moving animal, in the animal that are, using their brain under full operation, that we have not been able to see before. And it is actually in these minute details where some important changes in neural networks that also go along with malfunction of neural networks are to be detected, have to be studied. So I would very much say that we've kind of found anchor points for research that is starting now that will eventually lead to really novel discoveries.
To Kavli, yes, it was really an honor to work with Kavli, and I've been visiting themselves and met the key people there, and they actually was also very thankful for our support. But as I said in the beginning, it is also for us and my team extremely motivating to be able to contribute to this kind of important research. It's really a motivating factor, which is significant, I have to say. Okay. The Mini 2P solution you have leveraged kind of our TLens tunable optics, enabling optimal microscopic imaging across multiple planes. What would you say is in a way, can you explain the role of tunable optics in your device, and why is it important?
Well, yes, maybe without going into the physics of two-photon imaging too deeply, I can tell you the following. So when you look at a picture in a newspaper or on the screen, then what you see as one picture in reality consists of a million little dots, all side by side together forming that picture. Now, a brain explorer microscope, such as the Mini 2P, is in fact limited to only seeing one single of these dots at a time, not more. Therefore, to get the whole picture of brain activity, this brain explorer microscope must take 1 million successive single dot recordings. And when it looks into the depth, it must focus correctly at each of these single dots. And this happens at nanosecond scale, isn't only possible with a micro-adaptive optical system, such as your micro TLens.
So bringing those kind of single dots together into focus extremely fast clearly seems to be a critical kind of component of capturing the whole picture of the brain activity. Focusing, or should I say tuning, those individual dots into kind of a good image enables Mini 2P systems such as your PhenoSys product to deliver what they need to in a way make the researchers focus on the data and the data gathering and analyzing that, and very quickly can enable and set up your turnkey system into something very efficient tool for their research. So what about kind of, we are always thinking about what is the next, what's the next to come. So when we get kind of into a product with our technology, we always kind of day before that's the fact we start to think about the next version. What do you think is the next evolution of kind of Mini 2P solution, and how will that look like?
Well, to be honest, I'm not asking myself that question at the moment. I'm sure that people like you will come up with amazing technologies already in a few years. But from our point of view, with kind of the new technology that's just arrived, we have a decade of work ahead of us, I think, that we can really do
now. Okay. So we have minimum 10 years together.
I hope so, yes.
Okay, that's good to hear. So if we now want to kind of experience the PhenoSys Mini 2P in action, what is the best way to do so?
Well, great that you ask, Oyvind. So the next opportunity is actually just around the corner. It's the Society for Neuroscience 2024 meeting in Chicago, which actually draws tens of thousands of neuroscientists from all over the world, starts October 6th. And we will have our turnkey Mini 2P demo system available there at our booth. And we'll have experts there that can gladly consult interested scientists, show them the system, explain them what new things you can do with it. Our experience, and it's very necessary because our experience shows that such novel and innovative technology, disruptive technology to some extent, takes a bit of time to be adopted in the market, to be adopted by the people who will then use it in the future. Hence, we are currently running also several demos, for example, at the Zuckerman Institute in Columbia, at the Max Planck Florida Institute. And ahead of us is also a larger US roadshow planning to start in Q4. So plenty of opportunity for those interested to get in touch and see the technology in action.
Super interesting. Jörg, thank you so much for having this conversation with me, sharing your thoughts, your insight of the importance of this research and instrumentation, and the critical role imaging plays and tunable optics. I appreciate very much your time, Jörg. Thank you.
Well, thank you, Oyvind. Thanks is also on my side. So science is always grateful when industry also cares about highly special niche markets where your sales may not be so high and your profits not huge, but we will do our best that humanity will, I think.
Thank you. Thank you.